下，王夏博士呢，以前是 the former director of next-gen sequencing and bioinformatics at Baylor Genetics, an assistant professor at the Department of Human and Molecular Genetics at Baylor College of Medicine. His research interests include the discovery of the novel disease-causing genes, in-depth interpretation of next-gen sequencing data, and molecular genetic studies of eye diseases. He has published multiple peer-reviewed articles in the high-profile journals like Natural Genetics and the American Journal of Human Genetics and Genetic Medicines. He is really a productive and researcher and an excellent molecular geneticist. So we'll welcome Xia to join us at ACGA virtual seminar series. Xia, take it away.
Okay. All right. Thank you, Dr. Li. First, I would like to thank you and the ACGA for organizing this seminar. I think it's a great educational activity for the members and all the geneticists in the field. And I want to thank everyone for taking your weekend time to join this seminar. So it's my great pleasure to be here. So today I will talk about clinical exome and genome sequencing. In the first part of my talk, I will discuss the so-called analytical validity of exome and the genome in the clinical setting, mainly talking about their different capability to detect different variant types. And then in the second part, I will touch base on the clinical utility of the West and whole genome, including the change of clinical management and cost saving. So as you all are very familiar, clinical exome sequencing was launched in the US around 2011 or so, at Baylor and a few other labs. Ever since then, a lot of large clinical exome cohorts were accumulated, and a lot of the data analysis have been published in very good journals, such as two important papers I always want to brag about, by Dr. Yang's group. The first 200 cohorts at Baylor were published in the New England Journal of Medicine, and the first 2,000 cohort was published in the JAMA, discussing about the general finding of this clinical exome cohorts and the workflows, the SOPs, the standards to analyze this data in the clinical setting. And then around the same years, I think some other groups, such as Steve Kingsmore's group at the Radius Children's, they published their finding from the clinical whole genome sequencing in the clinical setting, mainly in the NICU setting. And they really emphasized the very short turnaround time of their tests, the so-called rapid whole genome tests. And really, those studies also discovered and showed us the clinical utility of these tests. How does it change the clinical management, and so on and so forth. I will touch base on that in a little bit more. So I was asked a lot about the question regarding whole exome genome, which one's better, which one shall we use? I think that depends on a lot of factors. But for me personally, I always like to see really if there's any diagnosis rate differences between the two types of tests. Obviously, if you look at the papers published by different groups, they're coming from different cohorts, different pre-existing testings were done, yes or no. So the diagnosis rate varies, but this paper in 2018, they summarized a lot of studies from either exome or clinical exome or clinical whole genome. They summarized their diagnosis rates. For whole genome, the average is about 41%, and for whole exome is around 36%. And they did a statistical analysis, and their conclusion is that there's no statistical significance in terms of diagnosis rate. Of course, this is a study published 4 years ago. I believe lots of things maybe changed, and people may have different conclusions from their own studies. But in general, I think the diagnosis rates so far are comparable between the two type of tests. And before we dive into the technical details, I just want to remind everyone that one of the biggest differences between whole exome and whole genome is that for the exome testing, there's one step to, yeah, somebody may want to mute themselves. I think we're hearing what they say, but for whole exome sequencing, there's one step to really enrich or amplify the region, which is the whole exome region, versus for the whole genome, there's no such step. And obviously the target region is different, and then because the whole genome doesn't have this step, there may be some I would say biochemistry or wet lab differences that may determine the different data characteristics in the later stages. So this is the data from our iLife. We were trying to identify the SNP detection, SNP detection accuracy. So it's single nucleotide variations or small insertion deletions by either whole exome or whole genome. We are using the data, benchmark samples from Genome, the Battle, and NIST. We calculate the sensitivity and specificity for either whole genome from BGI, whole genome from Illumina platform, or whole exome from Illumina platform. In general, their sensitivity and specificity are quite comparable, basically. But I just want to emphasize that sometimes whole genome may have better coverage in certain conditions. For example, this slide is cited from this study in 2018. What they found is that for whole exome sequencing, the relative coverage shown in blue at different GC percentage is different, and generally it increases from 30 to 50%, and then reduce dramatically when the GC percentage increases. And if you look at the data from a whole genome, which is much more smooth, so the changes of coverage by different GC percentage is not affected that much or dramatically as the whole exome data. And then on the right side, basically, here's a example showing you that on the upper panel, there is a deletion, small deletion in the RPGR, open reading frame. No, I'm sorry, in the GUCY2D gene, it's a retinal disease gene at the region. It can be detected by the whole genome, but it's actually not even well covered by the exome. So there are some coverage differences at when the GC percentage is very high, the whole genome may have some better coverages. I'm just showing you another example from our lab that's in one sample. This is a whole exome sample, mean coverage of 113. This is captured by one company. And you can see that in this disease gene PTBN11 Noonan disease gene region, this location, there's only one read. So it's not well covered at all, although the mean coverage is 113 versus another whole genome data with the mean coverage of 36. If you look at the same loci, the coverage is actually 55, which is quite high. And if you see the gray bar shows the coverage across this region, they're quite uniform. That doesn't change too much, across the intron and genome. So I think sometimes whole genome may have more uniform coverage, and that may make a difference if it happens to the different regions happens to be a disease-causing region. The other thing for SNP detection is really the non-coding variants, right? For whole exome, by definition, it only covers the exome region, and the probes may extend a little bit into the exome intron flanking regions, but it doesn't cover the deep intron regions or some other UTR or even intergenic regions. there are some version of clinical exome tests which have enhanced the probe design for those non-coding mutational hotspot region. For example, CYP290, there's a very well-known deep intron variants, and ABCA4. There's a lot of other examples, which if you know there's a mutational hotspot, non-coding region, people can really add on the probes to improve the coverage for those regions. So they call it enhanced probe design. Versus for the whole genome, actually it covers all the coding and non-coding region, right? But what we found when we analyze this whole genome data for this non-coding variants is that there are just too many non-coding variants, non-coding candidate variants for you to analyze it. The biggest challenge is that if you assume that some of these non-coding variants are disease causing, how do you prioritize it? How do you identify the ones that are clinically significant that really is the disease causing variants? It's very challenging. Even you apply different filters, like you're trying to match the disease phenotype, right? You're looking for de novo variants in autosomal dominant disease gene, for example, or you're looking for biallelic variants in autosomal recessive gene. Still, there are just too many candidates for you to analyze it. There are a lot of studies published which basically those a lot of tools, in silico prediction tools were developed, particularly for this whole genome data analysis, such as CAD, RAM, primary AI, and a lot of other deep learning algorithms. They always claim that they have very high accuracy in terms of prediction, the pathogenicity for non-coding variants, for genome-wide variant analysis. But in reality, when we analyze these variants, for example, using ACMG evidence, these predictions, most of the time, we only consider it as a supporting evidence, right? PP3, for example. So it doesn't make too much difference for our clinical analysis. So it doesn't provide a very strong evidence for us to, for example, rule out a variant. So we found I found this genome-wide prediction tools are very useful, but they don't help that much. On the other hand, a lot of studies propose that in addition to the whole genome sequencing, we can always couple it with other omics analysis. For example, RNA sequencing may help to prioritize variants that affecting the splicing. So basically, at the beginning, we can still apply the traditional strategies to filter variants based on their genetic evidence, based on their co-segregation with the disease, biallelic de novo, based on the phenotype match. There's also a lot of existing algorithms, as I've just mentioned, that have relatively good prediction power for splicing changes, such as SSF, MoxENT scan, and Splice AI, and more, to name a few. And then once you identify these candidates, non-coding variants that can affect the splicing, we can correlate these DNA variants with the RNA-seq data, for example, from the affected tissues, and looking for changes that's associated with alternative splicing events from the RNA-seq data, differential expression or monolithic expression, and more. And then once you identify these variants, lastly, you can further confirm it, by RT-PCR, or you want to pick up the variants and put it in the mini-gene assay to see if it really, in a well-controlled environment, if the variants really change the splicing. So this is in my opinion, that this strategy can provide you more solid evidence to prove or disapprove that if a non-coding variants can really have some functional impact, for example, affecting the splicing change. So this is basically the so-called multi-omics approaches, with the genetic data, with the DNA variants data, and then together with other omics data, either from RNA sequencing or from proteomics study and some more. and then trying to, with rigorous statistic analysis and trying to correlate the DNA variants with the functional output to identify the cause variants, and then perform further functional study if needed to prove that the variants are really the disease causing one. These strategies are actually well used, when people are trying to identify novel disease causing genes. But if you want to apply the strategy in the clinical setting, I think we need to have assays that are high throughput, short turnaround time, and cost effective. So there are more challenges to really apply these concepts into the clinical setting. Then let's talk about the copy number variations. So copy number variation, CNV, when we use whole genome to detect the CNV compared with the whole exome, because there's no capture or enrichment or amplifications, as I've mentioned, the coverage is generally very uniform across the genome. And I think it's easier to identify the copy number change signal comparing with the exome data. So I'm just showing you an example. This is a whole genome at very high coverage, 46x. There is a 2.25 megabase heterozygous deletion, which is very clear when you compare the copy ratio from that deletion region to the neighbor region. The difference is very clear if the signal are well normalized. And even when the deletion or CNV gets lower, you can still very easily identify these changes. This is a 8.5 kb heterozygous deletion TBX4 gene. And then there's usage of so-called low-pass whole genome. I think probably Elvis is in the audience, and he's really expert on that. For the low-pass whole genome testing, at basically using the whole genome testing at very low coverage, people can identify all kinds of different aneuploidy or CNV with no problem. This is showing you a very small duplication that can be identified at a low-pass whole genome 2.8x. And sometimes at our lab, we even identify single exon deletion using low pass. Of course, because this is your gene in the X chromosome. If you lose one copy, that's hemizygous deletion. So at that region, there's basically no probe. So it's relatively easy for people to identify that. But nevertheless, I'm just trying to say that with low coverage whole genome, you can still identify something that is very small. For exon, CNV exon analysis, as I've mentioned, I think the challenge is that the CNV cause, they're very noisy. I will show you the data in a bit later. Their coverage is not that even across the genome, likely due to the one additional step of enrichment of the whole exome region. And when you compare with the panel, I think maybe because there are way more targets and probes to capture, and there's competition between them, the signal is also noisier than the panel. And for panel, typically you will get higher coverage when you generate the data. Also for panels, because the panel size is smaller, for whole exon, you will have less co-capture samples, to sequence at the same time for you to do the normalization. So it's very challenging, I think, in a sense, when compared with the exon and the whole genome. But people are doing that in the clinical lab. So here are some of the CNVs that, just for examples, showing that we identified in our lab by exome sequencing and further confirmed by different orthogonal method. For example, here we're showing you a 2.9 megabase deletion of the DeGeorge region. We not only call it from the proband, we also compare it with the father and mother, so we know that it's a de novo deletion. And then this is the plots that we usually get from our system from the exome data. The y-axis is copy ratio and the x-axis is the genome references. As you can see, the signal looks much noisier. Each bar is a signal from one exon in the region. And you can see that, even we called a deletion in this region, it's a big deletion, but the signal is, in my opinion, is bumpier than the whole genome data. And the variations at different exon can be very high. So that makes it, I think, challenging to identify these deletions. Here is another example. It's a 2 exon deletion from HPS1 gene from the exome data. In this case, I think the bar is much more apparent. The pattern is much more apparent that these 2 exons have really around half of the copy ratio of normal exons. And then sometimes if you're lucky enough, if the deletion happens near the exome region or in the region that can be captured by the exome probes, you can identify the junction reads, and that will help you to accurately map the deletion breakpoints and kind of confirm the redepth analysis you identified for the deletion. So in this case, that we are relatively sure that these deletions are real. And lastly, I'm showing you a single exon deletion. In the upper panel is the single exon deletion. In this exon, it's heterozygous deletion versus a normal non-deletion case in the lower panel. Basically, I'm just trying to say that you can identify, even for a whole exon, we can identify single exon deletion. But of course, the challenge is that there are lots of candidate deletions for you to do the analysis. So it's very challenging to reduce the false positive rate. Okay, so next time I'm going to move to the structure variations. So for the structure variations, usually people say that they can be identified by whole genome, but it's also challenging because if we are talking about the short read sequencing from the Illumina or the BGI platform, as you all know, the short reads, they have intrinsic limitation in detection SV with high accuracy. You would identify a lot of them, but it's very hard to confirm, and a lot of these signals will be false positives. Here is one example that we identify the structure variation translocation basically from a NA sample with a known translocation between chromosome 17 and chromosome 3. And this is whole genome data, and we are able to identify not only translocation, but the exact breakpoints because we are able to find these junction reads, and we are able to map it to the 2 translocated chromosomes. So breakpoint is very clear in this situation. But there are studies suggesting that a lot of the structure variations that are identified by the short reads method is actually, those calls were actually unique to the technologies. So basically means that a lot of these calls are actually false positives. A lot of these unique calls, unique to the short reads calls, they were called as translocation, but if you analyze it using long reads technology, they were actually insertions. So here is an example from this published study in 2018. In the upper panel, it's called as a translocation by short reads data, and then they further use PacBio and Nanopore, and they show that this is actually an insertion, not a translocation for some reason. And on the right side, it's the same example. I think in the short reads platform, they called it as an inversion, but then they performed the analysis using PacBio and Oxford Nanopore, and they found that it's actually insertion. So it's the accuracy of SV calling by whole genome sequencing, if using the short reads platform, is still not very high and could be a lot of false positives. And then another layer of difficulty is the clinical interpretations. If you identify the structure variations, how would you interpret them? If it's unbalanced structure variation, usually, unbalanced means that there's a gene dosage change, and then you would identify copy number variations, and that actually, make it easier for you to analyze a CNV. But then if it's a balanced translocation, and then that really depends on where the breakpoint is, whether the breakpoints affect certain genes at all, certain diseases. If the breakpoints directly disrupt the gene of interest, then that's actually a very good finding. And if the gene and diseases fit the phenotype, that's great. Actually solve the case. But if the breakpoints doesn't affect the gene directly, it breaks some non-coding region, or intergenic region, then it's more complicated. It may be it'll affect the 3D structure, or those long-term effect elements may affect those type of effects, but the effects is less obvious versus if the breakpoints actually land on a certain genes. So I think there's another layer of complexity to clinically interpret those structure variations. Okay, then now move on to the short tandem repeats. So for the short tandem repeats, here I'm summarize this is from a paper published last year, summarize the currently known short tandem repeats and they're in the disease genes. As you know, the STR may happen either in the coding region in the exome. For example, the Huntington is caused by the polyQ repeats in the coding region. It may also land in UTR region, for example, Fragile X, 5' UTR, or in the intron region, like Friedreich ataxia and more. So obviously, if using exome to analyze, you would only able to analyze this STR from the coding region and some of the UTR region, but not the deep intronic region. But overall, for whole exome and whole genome, I think the biggest challenge is still that because it's a short reads master. If we're talking about short reads master, the read length is too small. Sometimes it's cannot accurately call the STR repeat numbers. I want to point out two papers that recently published. Those are big cohort study regarding the analysis of STR from either exome or whole genome technology. This paper talks about the analysis of STR from the exome cohort. They basically study their cohort is they basically apply whole exome sequencing for two cohorts. In the first cohort, they have over 2,800 patients with movement disorders, which are associated with lots of known STRs. This is called cohort 1. And then they have another cohort that's, sorry, let me focus this. They have another cohort over 11,000 patients. Oh, no, not this one. Another cohort over 35,000 patients with other disorders. And in both cohorts, they apply the whole exome sequencing. And the methodology is they use the expansion hunter as the STR caller, together with or without manual curation, to inspect the pileup of the exome data, and then plus the PCR confirmation. And then what they found is that they can confirm 34% of STR cause from the first cohort. And they can confirm a bit more, 56% of STR cause from the second cohort, with the help of additional manual curation. So basically, they call the STR, from all these patients, and they are able to confirm anywhere between 30 to 60% of the cause, and the other cause cannot be confirmed. And so that's the specificity of the STR cause from whole exome sequencing. from this cohort. And overall, they are able to increase diagnosis rate by 0.2% by addition of the STR analysis. The next paper is published in this year in the Lancet Neurology. In this study, they used a whole genome analysis. This is the study from UK. Last one is from the Netherlands. From this UK study, they also have two cohorts. In the first cohort, they have about 404 patients from the National Health Service from UK. All these patients have previous PCR tests that are done. So those are gold standard cohort. And then in the second cohort, they have over 11,000 patients from the 100K Genome Project, and they are considered kind of like a prospective cohort. They use very similar methodology as the first cohort, Exome cohort. So they call the STR using expansion hunter, again, the same algorithm, but it's from the whole genome data. They will do the visual inspection, same as manual curation. Basically, they will manually check the BAM file, look at the PowerUp file, and they also do PCR confirmation. For the first cohort, the accuracy is surprisingly high. The sensitivity is 97.3% and specificity is 99.6%. So in this first over 400 patient cohort, they were able to achieve very, very high accuracy in terms of the STR analysis. But then in the second one, for the prospective cohort, they are able to confirm about 84% of the cause. It's lower than the first cohort, but it's much higher than the Exome cohort. But they also found that because they're doing short reads analysis, they found the lengths of the alleles that are larger than read lengths, for example, 100 base pair or 150 base pair, the lengths will be underestimated. When the actual repeat number or the actual length of the STR is larger than the read length, that repeat number will be underestimated by this method. So that's a limitation of the short reads, I believe. So as a brief summary, in terms of the different detection, powered by either exome or whole genome sequencing, in terms of the coding SNV, I think they are quite comparable. For the non-coding SNV, whole genome obviously identify a lot more variants, but the red one is the so-called clinical interpretability. So it's hard, it's very challenging to really interpret it and to identify the causal variants from those additional non-coding variants. For copy number variations, because the whole genome has very uniform coverage than the whole exome one, so it may identify more, but so far our experience that it's quite comparable between exome and whole genome. For structure variation, obviously, if the breakpoint is located in the non-coding region, WJS may capture more, but again, the interpretability is the bottleneck, or it's very challenging to interpret that, in my opinion. And then for STR, theoretically speaking, the two assays should be relatively comparable, but turns out that whole genome has better performance, but the overall challenge is the short reads. So if the read length can be longer, then the analysis of the STR can be much better. So that's it for the first part of the talk. Next part, I'm gonna talk about the clinical utility for the tests. Basically, trying to see if the clinical management can be changed by the diagnosis. So there are a lot of studies reporting, the clinical utility of the whole exome, whole genome testing. For example, this paper published in 2017 by Lin and Meng and others show that clinical exome sequencing can make impact in clinical management in different aspects. It could be initiation of new subspecialty care. It could be redirection of care. Could be change in the treatment or diet, or the change in the major, or leads to some major procedures, including transplantation. So indeed, showed that the diagnosis can change clinical management. And then there are a lot of other studies have similar conclusions, and this paper nicely summarized these studies and showed that, anywhere between 17 to 27% of cases after either whole exome or whole genome study, there is an actionable item. There is a change in the clinical management. Therefore, they were considered as clinically useful. The other thing that lots of labs are emphasizing is their turnaround time. So they call rapid exome, rapid whole genome. And the clinical benefits are the rapid tests, quick turnaround time, giving back the results in a short amount of time can lead to early intervention, which reduce the morbidity and mortality, and can facilitate the end-of-life care decisions, and may reduce the very lengthy and costly ICU stay, which is a lot of cost. So I think it's, in a sense, it makes economic sense to have a diagnosis in a very short amount of time. The study is from, again, I believe it's from Steve Kingsmore's group. They utilize the natural language processing, automatic variant analysis, and others to launch these rapid whole genome testing, and they all the way to reduce the turnaround time to about 20 hours. From the sample to the reports, about 20 hours. Maybe there's some even new records published recently. I'm just citing a paper from 2019 that these whole genome testing can be done in 20 hours or even less, and that in several cases, because there's no any prior testing has been done at all. In several cases, the rapid whole genome test indeed leads to a diagnosis and can potentially lead to change of the clinical management. And then there are a lot of studies analyze the cost saving part of exome and whole genome testing, even the rapid version of this test. For example, this study in 2017 showed that additional cost saving from avoidance of planned tests and procedures and reduced lengths of stay are estimated to be around $424,000 per case. That's for a rapid whole exome sequencing. And there's another study in 2018 showed that rapid whole genome, in the 6 of the 11 infants, the changes in management reduced patient costs by anywhere between 800,000 to 2 million. So there's very obvious cost saving out there if you can reach a diagnosis in a very short amount of time. The other thing that I'm interested in is that the insurance coverage. So before this talk, I searched Google and trying to see what's the current insurance coverage for whole exome and whole genome test. And what I found is that generally nowadays exome test is conditionally covered by several payers in their policy. For example, these policy from United Healthcare, they say that whole exome is proven and medically necessary for certain conditions. For example, diagnosing or evaluating a genetic disorder when the results are expected to directly influence the medical management, when it makes a change, and clinical outcomes, and all of the following other criteria are met. So they have a lot of conditions for you to be able to, to cover the exome test. However, for United Healthcare, they say that whole genome is not medically necessary for evaluating any genetic disorder because they think there are other equivalent tests that are available out there. So currently, they don't think whole genome should be covered. And similar idea from ACGA, they say whole exome is conditionally covered, but the whole genome is not covered. So honestly, I think from the science and clinical perspective view, I think whole genome do have advantages in certain scenario, but I guess, if you want to take the cost into consideration, and if you want to take the opportunity cost, and if there are other alternative tests out there, yes, indeed, there are more studies need to be done to show that whole genome test is also make economic sense for the payers to cover that. So as a brief summary, I think for the exome test, currently, I think it's a relatively cost-effective option. Nowadays, a lot of improvements have been done for the clinical exome sequencing. And if you look at the diagnosis yield at this stage, it's comparable to other tests, to whole genome. For whole genome, I think it's a all-in-one test. I think studies from the NICU, from Radius Children's, other groups clearly show that if the test whole genome was done in a very rapid fashion, it basically covered a lot of different type of variants at the same time in a short amount of time. So it's an all-in-one test, no doubt. And potentially it could have higher diagnosis yield because it covers more type of variants. It may have better coverages, but I think the current bottleneck is that the interpretability needs to be improved. This could be many things. People could use multi-omics approaches. In the clinical setting, these still requires these technology to be developed in a high throughput and cost-effective fashion. Also, people need better genome annotation. There are a lot of algorithms that studied in the name of machine learning and AI that claim that they can have better analysis and understanding in the variants, which is great. And then variant database build up and variant sharing, I think that may also help improve our understanding of the genome, which may eventually improve the interpretability of the tests. And then there are a lot of studies out there clearly show that for both whole exome and whole genome, especially the rapid version, they can lead to change in the clinical management in significant portion of cases in the clinical setting. And there is a great cost saving out there if you do it in the right way. And if the diagnosis can be made in a short amount of time. And then lastly, I wanted to say that I think because of the limitation of short reads, I think the long read technologies, they may have more technical advantages in detecting certain type of variants, for example, the structure variations, the STRs, even copy number variations, and they can also be done in a very rapid fashion. So there's great potential out there for those long reads technology, which can be complements for the current technologies that we are using, especially in the clinical setting. Okay, so that's it for my talk, and thanks for your time, and I'm happy to take any questions if you have.
Hi, Xia. This is Tao Xin. How are you?
Hey, Tao Xin, Dr. Huang, how are you?
Great talk. So
thank you very much.
For I have some comment, also some question. Comment is you see in whole genome sequencing, you can include the mitochondrial genome, but I think this thing include in the whole exome. Another question is you see when you do RNA-seq, the gene expressions are very tissue specific. And so many of a gene you only express a specific organ, like you see many of a metabolic disorder, because you see only occur in liver. You cannot have a liver tissue to do the test,
Right.
Okay. My question for you is this here, in terms of long, ideally you see use the long reading sequencing and also whole genome sequencing will be ideal, right? Of course, this is consider cost. And the length. So can you compare to me? Can you do a comparison? Use a different platform. Let's say you use Illumina short read versus Pacific Bio, and also you see the Oxford reading. So how the cost differently? Is a tremendous difference, or you think in near future, you think it will be get us closer? You will be very similar.
Okay. So yeah, Dr. Huang, thank you very much. Thank you for your very thoughtful comments. So I will try to answer these comments and questions one by one. So for the first one, I'm very glad that you brought up the mitochondrial. So for a whole genome analysis, indeed, we can regularly analyze the mitochondrial genome. When we analyze those whole genome data at a coverage of 20x or 40x for the nuclear genome, we found that oftentimes you can get maybe 2,000 or 3,000 coverage for the mitochondrial genome, which is a great benefit for the whole genome analysis. Versus for a whole exome, it really depends on the capture design.
Yes.
I know there's some designs that don't initially include the mitochondrial genome in the whole exome capture design, but later, people found it's important. It's better to be added on to the whole exome solution. So they do have some additional designs that covered both the mitochondrial and the nuclear genome. So I think that's a great point, that whole genome do have these mitochondrial genome material already covered in their data. The second one is regarding the tissue specific expression of the gene. It's very important for the analysis. I think what people was trying to do is that either they can directly get the affected tissue. In case they cannot, for example, what you were just saying, they can use iPS cells trying to induce into the particular tissue, but then obviously there's a cost and time out there. So yes, but I think it's very important to keep that in mind, that if you want to study the gene expression, you will have to study the gene, the affected tissue. That's definitely something important in the analysis and research. Regarding the cost comparison, if I understand correctly, Dr. Huang, you were saying trying to see if in the future, whether the third generation technology,
Correct.
whether the
Correct.
cost is,
Yeah.
is
Would be cost
comparable to
effective.
the
Exactly.
Illumina platform?
Yeah. Yeah.
Okay. I think, I'm not an expert in the third generation technology. A lot of the audience, feel free to jump in if you have more understanding on that. My understanding is that currently the cost is a third generation, those long read technology is actually not that high. I'm just using Oxford Nanopore as an example, though their kits, their sequencer is not very expensive at all. Of course, their reagents, the running costs may be another story. I think for the third generation technology, they're quite good at structure variation, STR, CMVs, and previously at the beginning, people think that their accuracy in SMV detection is not very high. Therefore, you still need another platform, to do those type of analysis. But nowadays, I saw a lot of papers published by different groups. They're claiming that their SMV analysis also can provide very accurate results. So if they can do good job on both, on both ends, on different type of variants,
yeah,
I think their cost is not very high. To be honest,
that's what I feel.
Yeah, that's what I feel.
Because the costs come down a lot, compared to
exactly. Yeah, at least for Nanopore, about PacBio, I'm not quite sure about that, but yeah. So their cost is not very high, I think. Yeah.
Thank you.
Thank you, Dr. Wang.
Hi, this is Ann Tsai. Thank you for the wonderful talk. Fantastic, really.
Thank you very much, Dr. Tsai.
I just have two questions. Okay, so on the in those papers who commented on WAS and the whole genome, didn't create more EO, diagnostic EO particularly. They often said that other than you avoid the capture deficits, like lose mutation, and they always say that you will sacrifice on the depth and the coverage. It's actually not helpful. But is that continue to be a problem after we have all these? And the other thing, they always commented on the non-coding region. We don't know too much anyway. So even if you have a variant, you still don't know what that really means.
Exactly.
Right. So is that continue to be an issue at this point?
I think the interpretation is definitely still a bottleneck. I think, at least in our lab, we analyze, quite a lot of whole genome data, but for the non-coding variants, we found them out there. We are able to somehow prioritize and pick up some of them. But even you report it out, we just don't have other evidence to confirm that it's disease causing. It's different than you find a stop mutation, you find a well-known, well-reported mutation versus if you find a novel non-coding variants, we really need other pieces of evidence to support its pathogenicity, because there are just too many, and by chance you will find something that fits the phenotype, that fits all the inheritance. By chance you will find those things. We just don't have enough evidence to support that. That's why I'm saying, okay, maybe we should other multi-omics approach in a high throughput and cost-effective fashion to help us to understand it. Maybe there's some, like 1,000 genome and nomad, right? Maybe there could be a RNA-seq or proteomics version of nomad or 1,000 genome out there that help people to annotate those non-coding variants already. That may help our interpretation. But currently, I do feel that's still a bottleneck. The interpretation bottleneck. You can detect a lot. You need some powerful ways to help you to analyze in a clinical setting.
Totally. And I just happen to have a clinical diagnostic dilemma. I actually have a child who manifests biochemically like NKH. Okay, but we only find one gene changes on the cleavage enzyme. We can't find the other one. And then if you said that you want to use RNA-seq, you will have a scenario that change the splice size. So you could actually have the expression pattern that you can see. If it's a missense mutation, it still doesn't help, right? And then, so in that scenario, we are really worrying about non-coding region changes. Would exome help, or I don't know if long read, short read, whatever, if you don't know what the variant actually caused the problem, what would be the best next step after we do the exome? And then clinically, we just can't prove it at this point. Even though biochemically you prove it, molecularly you can't. So in a scenario like this, how do we handle that? What do you suggest?
In your scenario, I think it's, how would I put this? I think it's because you have a diagnosis, right? You have a relatively focused diagnosis. If you really, and you already find one deleterious variant, right? So we're just missing the second variant. In that case, I think it doesn't hurt trying to explore a little bit more into the non-coding region, or even into the long reads scenario, looking for structural changes. Because once you identify those candidate variants, first mile, turn right after Zaxby's chicken fingers and buffalo wings on the right onto North I'm sorry. First is that it doesn't, because you already narrow it down to one gene, you wouldn't find too much candidate non-coding variants or structure variations out there. You probably only find a few, you can, or a handful of those candidate variants. And then because you don't have that many candidates, I think you can further perform some functional study to see if there's any consequence. For example, you identify a few non-coding variants, you can really use mini gene assay, for example, to see if it really affects the splicing. So if it really narrowed down to one gene, I think it doesn't hurt to trying to explore a little bit more about different variant type. But I'm not saying that you should try whole genome. I'm just saying that for that particular gene, you can try to analyze the variant types that are not currently captured by whole exome sequencing. And that doesn't take too much effort to further, study those variants, in my opinion.
Okay, and then is there like commercial lab that would do that? Just do a very focused gene,
that's
like a single gene.
Right. That's, we'll have to check. I mean, we'll have to ask, all the colleagues in the field. So I guess.
So I have a case for this scenario. We have we identify one for new gene FDSR, and indeed the whole genome help us found the second variant in intro, and then we were able to use RT-PCR confirm. Indeed, that variant in intro indeed cause splicing site change. That's the one. This is a good story. Another case is very similar to what Ang just mentioned. We have a patient clinically look like a fibrous disease, and then all clinical site include the biochemical test look like you see fibrous disease, but you see we are not able to find the mutation. My suspicion is you see some very in intron, or you may be in a 3' or 5', and so that can course, because you see, fibroid disease is a treatable disease, you know that, right? But you can imagine the insurance company, unless you document, prove this is a genetic mutation, insurance will not cover the cost because that's a very expensive drug. So at this time, my approach is we try to contact people, you see, that are doing research, they're very good, you see, in this particular gene. And then then I didn't see any commercial lab can help. And unless you want to, of course, do whole genome sequencing will help, you know, so at least help you see that suggest some of the variant in intron you see in non-coding region, and then so that can help you to guide you. So, okay, what is the next step? But as you said, you need a second technology to confirm indeed that vary is indeed cause something, cause like splicing change, or you see regulatory region mutation, which is affect the gene expression. So this is always have some case like this is in clinic. It's a big dilemma.
Yeah, absolutely. I think, you know, both of your scenario, I think, you know, if you can narrow it down to one gene, you already find one copy and the phenotype fits, you know, then definitely it's worth to dive deeper. And, you know, as Dr. Huang just said, you know, there could be some other research lab out there. They already have a well-established battery of assays for certain genes in which they're expert. And then they can easily help you to study your variant of interest to see if, you know, any of those mutant construct will have some effects in their functional readout. So I think,
I have a couple comments. First, that I disclose there is no conflict of interest. So we recently had a couple successful story using the whole genome platform to focus on single gene test, which we already identified one variance and on the gene, but also fit the clinical presentation. One is the Wilson disease. So they're siblings. First sibling having biochemical abnormality. The second one still young and has a borderline abnormality. And so we find one variance on both. So we really don't know the younger one is that really affected, not and biochemically clear, or is just a carrier. So that's definitely changed the management of the younger sibling. And so we write under the medical necessity letter and we send it to Variantex. And so we just do single gene analysis. Actually, they found a very deep intron variance, and luckily that variance already reported in 5 unrelated cases. So it's easy for them to interpret. I think it depends on the case, and insurance still could consider, because it does change the management and it's changed the outcome. So that's something and you can try and the other thing, just thinking forward, and I know there are some link RNA affect the gene expression, and actually I think last year, Science published an article for PKU link RNA and affect the enzyme affect enzyme activity. And so I just don't know. And for protein coding gene, and you still can knowing they change amino acid for this RNA, non-coding RNA, even we identify variants, and what's the way and to functionally prove those? I feel that's definitely a big gap and for our knowledge, and I'm not sure what's the future and how to study those link RNA, non-coding RNA to prove their pathogenicity or function.
Right. So I think what I mentioned in the slides is that they call it allelic expression. Basically, what they're trying to say is that when there is a non-coding change, for example, even in the link RNA, for the mutant versus the wild type, when they check the target genes RNA-seq data and check the expression, they could be a different level of expression associated with particular, mutant versus the wild type. They call it allelic, expression. So there are statistic analysis that can correlate the two events. On one side is the DNA change, even in the non-coding region or link RNA, versus on the other side is the expression level differences from the RNA-seq data. And you can perform statistic analysis to say that these two events are correlated. And then you would need a orthogonal method or secondary method to confirm your finding, right? For example, if you think this is the link RNA change that really leads to the different expression, you would introduce these variants in an in vitro system, for example, something like imaging assay or designed for your variant of interest, and compare the gene expression between the mutant and the wild type, and see if it really causes expression changes, something like that. So you would need a primary finding, coupling the DNA RNA data and statistic analysis, and then after that you would have to confirm that in a secondary assay. That's my understanding. Yeah. So there could be different model in which the non-coding RNA, the non-coding changes could affect either different level of expression or abnormal RNA splicing or something more, and then you can further prove that in a secondary system.
Yeah, definitely multi-omics.
Right. Yes.
Yeah, the amazing is that link RNA they identified for PKU, PAH was not interact with RNA expression, was interact with a protein and affected the BH4 or Phe binding. So it definitely RNA, even you have a liver tissue and RNA-seq is not helpful in that case. It's just so many unknown and we don't know.
Right. Yeah.
Yeah.
Question for you, Hong. And you see for first case, you say that you found a deep intron mutation in a case with Wilson disease.
Yeah.
Did you say you do a whole genome or you see a whole gene for that particular gene?
So they use the Variantex use the whole genome platform and
whole genome profile.
Yeah,
okay.
and they just look at
Oh.
the data for that gene.
Yeah,
because we tell them.
Now, in that case, you see, you're pretty lucky, because insurance
Yeah.
cover for that. Because this is you see, you pretty much use a whole genome to looking for one particular gene.
Correct. So I just write the letter. And do you want to treat the sibling or do you want the? And then they approved. Yeah, it's a wonderful talk. Thank you for sharing all this information. I think this virtual seminar series and it's going to be held every other month. And if more and more people want to volunteer to share their work, and we can definitely change it to monthly. Yeah, thank you for joining us. Yeah. Thank you, everyone.
Can I ask another question?